is Shahani, and I am part of technical services team at Oxford Nanopore Technology. I'm with the company for past 2 years, and before that, I was with Columbia University Medical Center, working there for a while, like I think from the middle of my PhD to postdoc, and then as a senior scientist, I worked there. The way this advanced training will take place. So first, I'll give you a very brief introduction of Oxford Nanopore technology, like how does it work, and then what type of library prep we are going to do today, and maybe a little bit about P2S because that's the platform you have in your lab. And then we should start, planning our day, because they are like reagents, we need to thaw it, it takes time, and then the flow cells, it has to be at room temperature. So there are as the experiment needs to be planned, we can also plan based on the QC of the samples, which you have already done, which would be better candidate for today's pilot experiment. And it's because the library prep which we are going to do, it's NBD, which is a multiplexing kit based on ligation chemistry. It takes a little bit longer time than our speed optimized chemistry. Like we also have speed optimized chemistry, which takes actually a few minutes to make a library prep, So there would be some gap because there are PCR steps and incubation, and we'll come back and again understand a few other topics related to Nanopore, example, let's say accuracy, or what are the different other library prep you can do using Nanopore platform. Little bit about P2 Solo, how we can do software check, or what is the software, how the base calling is happening, and then about sequencing, how we can do the sequencing, how we can monitor the run. So today it would be very preliminary, tech talk regarding the Nanopore chemistry and the library prep and what we can do with this platform, and then. At the end of the today, we'll have some idea how the sequencing run is taking place, and based on that, we can plan tomorrow. So tomorrow, I think I will go more on the analysis side, or also how we can interpret the sequencing run itself, or specifically for your lab, you will be dealing with cfDNA, what you can do regarding that. And then, we'll see how the run is going on, if we need to do some kind of wash and reload and everything. And then day after tomorrow, I will invite our bioinformatics scientist. He can actually show you or talk more about the bioinformatics part. So this is how I planned. And, let me know if you want to change anything, if you want to discuss something else, we are happy to do that. Let's start. I think this. Okay, so as you may already know, how Nanopore sequencing works. So at the center of Nanopore lies a nanoscale hole, which is a protein molecule, and that is called nanopore. And it exists naturally in different biological systems, example, across membrane, So what we have done at ONT, or Oxford Nanopore Technology, we have enhanced the property of naturally occurring protein by introducing some mutations across the barrel region of the nanopore, and then embedded it across lipid membrane. So in simplified words, I would say nanopore is like a molecular Coulter counter, which we often encountered in our biology, like high school, college, we have encountered it multiple times. So to understand how exactly this technology works, let me put this pointer here. So here you can see this blue molecule. So this is the nanopore, and it is embedded across this gray colored lipid membrane. And also you can see there is a purple, macromolecule. So this purple macromolecule is called adapter protein or motor protein, and it has helicase property. So what it is doing when, let's say, DNA strands is moving through this nanopore, it helps to unwind the strand. And not only that, it is also controlling the speed of movement of DNA or RNA or the nucleotides which are going through the nanopore. So in DNA space, that speed is close to 450 bases per second. And in RNA space, it is close to 120 bases per second. Also, I think it's worth mentioning between DNA sequencing and RNA sequencing, this protein is not the same. It is different. Like one helicase protein is one motor protein is specific for DNA versus we have a different type of protein specific for RNA. And also the flow cells are different. So when you do DNA sequencing, it's DNA flow cells. When you do RNA sequencing, it's RNA flow cells. It's very different. So what's happening actually here, like there is an electrical potential. It is getting applied across this lipid membrane. And due to the presence of the electrical potential, DNA or RNA strand can move through the pore because, it's because of the negatively charged molecule which will move when there is an electrical potential difference presence. And what happens when it moves, it generates disruption in the electrical energy. And that is called squiggle, you can see here. And in nanopore system, this electrical energy gets deconvoluted using our machine learning based algorithm or software. And because of that, we are able to know information of each and every base at single nucleotide resolution in real time. I will go over this base calling mechanism or like how our software is deconvoluting the signal more tomorrow. It's like, I am using very simplified words, but it is actually a very complex algorithm and a very highly trained model what we use. So we don't need to understand everything if we are not part of a software development group, but I will show you like at least the basic pipeline, how the base calling model works.
I have a question. So
Sure.
how exactly does the motor enzyme identify the DNA or RNA strand? Like, is there a specific region that you attach with your library, or how does that?
So I will actually talk a little bit more about it. So, in Nanopore, the library prep is actually means when we are adding the motor enzyme. That's the library prep. And I think in the next slide, most likely it is more clear. So this motor enzyme, it also has an adapter sequence, which is a single nucleotide sequence, and that is acting as a tethering molecule. And it helps to bring the DNA or RNA close to the nanopore. And then, electrical potential is applied across the membrane, and it is going through the membrane at a particular speed based on, whether we are doing DNA sequencing or RNA sequencing. And then because of the presence of our software, which is called MINO or the base calling software, we are getting the real-time sequencing information. So what are the main features of Nanopore? As you can see here, like it is a direct sequencing of DNA or RNA platform. We are not introducing any kind of PCR steps. And because of that, this information are like the entire data set is free of PCR biases. And also because of that, we are able to see the information of base modifications using a Nanopore, like any kind of sequencing system. Um, like any kind of methylation signal, if you're interested to explore, it can be done using nanopore chemistry, and you will get the data in real time. So when nanopore, like ONT, came to the biotech world, that time it was only a long read sequencing platform, but presently, because of all the improvement happened in the past 10, 15 years, like this is a read agnostic platform, which means we can sequence as low as 50 bases when we are thinking about DNA sequencing to as big as few megabases. So the record is 4 megabases long sequencing, but that is like not a median sequence. Like N50, you will see median N50 is way less than a megabase. And yeah, I think the great benefits of using this platform is real-time analysis as well. So you will be able to see the base calling information in the real time. Also, it's a very scalable platform. I mean, you have P2 Solo, but in a Nanopore community, you will see like we have a platform which is as small as like iPhone. It's called Minion, and so it's very scalable. And we also have P24, which is like the same PromethION flow cell, but you can run 24 slots at the same time. So that is our largest platform. So yeah, here, like, this is how the R10 sequencing chemistry works. Like, example, here, this is the motor protein, which has a like adapted sequence, and because of the tethering, it is helping the DNA strand to come closer in close vicinity to the nanopore, and it helps in the movement of the native DNA or RNA across the pore. So today, like we are gonna use a native barcoding library prep chemistry, and this is a part of our ligation-based chemistry. So what we are gonna do today, and I will once again, when we have the gap, I am gonna go over other types of library chemistry, which you can use later. Like later today when we are talking more about different types of chemistry. But now let's focus on native barcoding chemistry. So example, the sample, like the starting material would be gDNA. In your case, it's like cfDNA. And then if we use gDNA, then fragmentation is suggested. Because we can do without fragmentation as well, like this is an optional state, but we have observed if we do fragmentation, then there is less pore blocking, we get more data. And then the way this chemistry works, actually, we are going to do some kind of end repair, and then we are going to do barcode ligation based on like how many samples we want to pull and everything. And then we are attaching the sequencing adapter. And that's actually what the library prep would be.
When you have fragmentation, right? Because so in bulk sequence you have many transcript of the same region. So would it be something like that here, or
no, you are talking about short read sequencing.
Yeah.
So short read sequencing is very different, right? Let's say you are using Illumina, then the fragment length, the maximum you can get, I think it is close to 250 or 300 base pairs. If you use NextSeq, which I used for a while, I think it was 75 base pairs, bases, So that's a very different chemistry. We are not, fragmentation is not for, when I said fragmentation, it's not RNA though, it's gDNA.
Okay.
And it's because, gDNA is like really long thread, right? So you can do without fragmentation, that's fine. But what will happen, there could be pore blocking. Like, maybe you lose all the pores faster than if we do fragmentation. So if there is a sweet balance between what we want to do or what fragment length we want to think about, but it all depends on what type of project we are doing, what are the starting materials, is fragmentation is necessary, what is the data output we are looking at, So we do also have one chemistry, it is called ULK, ultra long chemistry, which we do, example, when we were looking for de novo assembly of genome. And for that, we need really long length of N50. So there is no fragmentation. It's like you have to handle the sample a really gentle way so that there is no shearing of the DNA, so that you can achieve, like, let's say 100 kb or more, length. Because the reason is, if you don't do it, then when you are doing the de novo assembly, you have to stitch it computationally, and we don't want to do it because in our platform you can actually get really long length of N50. But that is not applicable for cfDNA.
But what if there is because suppose I extracted cfDNA, but I know I have genomic DNA contamination. Do you still fragment it or no?
Because even if you fragment it, it's not about fragmentation that time, right? Yeah. It's like, can you purify genomic DNA if you fragment it? The answer is no, because it's still part of your pool, right?
Yes.
So when you are sequencing, you will lose data to genomic DNA, because fragmentation is not a purification step. It's absolutely just to get more data out of your sequencing. Right?
Yeah. So what I would be concerned about is if I don't fragment and if I'm looking for cfDNA, it will block my pores, right?
It depends. Yeah, if you, for cfDNA, the protocol, the way, what I said, it's very specific for gDNA sequencing. I'll show you what our recommendation for cfDNA.
Okay.
But also at the same time, I want to mention all the benchmarking data we have. It's from plasma cfDNA.
Okay.
We do not have any internal documentation which can support CSF cfDNA.
Okay.
So if any optimization is needed, that is something you have to do. We would definitely help you out, but presently I don't have any data which I can share, which will help you to get rid of the gDNA. For plasma DNA, we have not observed what you are getting. Yeah. So I already, shared that piece of data, not data like the QC, which Teddy shared yesterday to our internal team who are working on cfDNA. And, I'll let you know if they give me any kind of specific ideas like towards CSF, cfDNA, but presently I don't have. So this is like the Promethean flow cell, which you are going to use today. So Promethean flow cell, I will once again show you like when I see that flow cell physically, but how this is just to give an idea of what we are going to deal with. So there is some connector, which means it's going to give us the connection with our platform, which is the P2 Solo and this Promethean flow cell. There is always some buffer inside the flow cell, so we can get rid of those buffer using this by just pipetting out from this west port. There are two ports present. One is here, one is there. Once again, like you will be able to understand it when we do it more. The picture is not that clear. this is like waste reservoir because, as we do the sequencing, you will see like accumulation of used like reagent or buffer here. So these are the reservoirs. So we have some reservoirs here where it would be accumulated. The important part, which is the sequencing chip, it is here. And there is a sample input port. So I will show you how to load a sample. It's quite easy, but we have to be very, careful. Like when we are doing priming the flow cell or the introducing sample, there should not be any kind of air bubble get introduced during that step, because if there is an air bubble, then that sequencing is not going to work efficiently. But we do have, ways to do that. I mean, it's quite simple. When I show you, you'll understand what I'm talking about. And especially for ProMethane flow cell, I have never seen like, air bubble is getting introduced unless we are forcefully doing something, mistake. Then the bubble would be there. So it's very user friendly. It's like sometimes, from the slides, it looks like it could be sometimes overwhelming, but when you do it, you'll understand that it's very user friendly. Okay, so I think we will start, working on our experiment part, but I think I just want to quickly share something about the P2 Solo. So this is the platform you have, P2 Solo. So there are like two slots here, right? These are the, it has this, where we are gonna insert the flow cell. And there is a LED, like a type, like light there, like you'll see when the sequencing is happening, it's kind of green and blinking. If there is an error, it turns like red. So these are the indications also whether it's working well or not. But I mean, this is a good indicator, but I usually check the software, like, some whether it's going well or whether some error message is popping out or not. Yeah, so there are two ways to see it. And it's there is an USB-C powered device, like there is USB connection at the end. I
have a question.
Sure.
What's the difference about this model and the Mint Ion? Because I used the Mint Ion before.
Oh, okay.
And that one did get a few air bubbles.
Yeah, so the chemistry is same, it's the nanopore chemistry. But the flow cell or the platform is completely different. So let me see if I have a Mina flow cell photo or image here. Like the way you load or way you prime the flow cell, you load sample, it's a little bit different than, and it's because the flow cell is completely different there.
Yeah, so it looks like a car, like a round car.
It's like more like a rectangular shape, and it's way and the way we take off, let's say, buffer or do the sample introduction is different because physically it is completely. The microfluidics, not the microfluidics, the engineering of the MinION and the ProMethane is different.
Okay.
And it is possible because MinION came first, and that time probably, like the Nanopore, like the engineering team, they made that flow cell that way, that's how it was built. But when the ProMethane came, that engineering part of the flow cell became completely different. So it's only the structure, I would say, of the MinION is different from ProMethane, but the chemistry is same. Sure. Everything is same. Okay. Yeah, the software is same. Those are not high output flow cells. So if we are dealing with whole genome sequencing or even for RNA, those are not the best platform, I'd say. I mean, you can always use it, but the data output would be less. And then, and for some labs, probably that is enough. But usually for whole genome sequencing, when we do analysis, we want deeper data, right? So that will not, you won't get it from MinION. But for microbiology labs, virology labs, I think MinION is enough, yeah. So I think you already did that, but this is it suggests like for P2 solo specifically, what are the major steps. So when I think it was done, I think Teddy or someone in your lab already did it. So when the platform arrived, it also comes with something, some flow cells called CTC. So these CTCs are very difficult to replace. So just make sure like you don't lose it, because if it is lost, I mean, definitely there is a way to replace, but it is coming from UK, so there could be delay. And these CTCs are very useful because for hardware check, or even let's say there is a software update, we do the software update, and then using these CTCs once again we make sure like everything is working well or not. So, and what else? Like, there is like USB Type C to Type C cable into the P2 Solo USB Type C port. And they say this is very important. The P2 symbol should face down. So these are the things with P2 Solo. Like we have a very like not similar means we have also another platform called P2I, where also you can use like two flow cells at the same time. But for P2 Solo, there are some additional criteria which we need to take care. Like we need to make sure the USB is like the P2 Solo symbol is facing down. We need to make sure the computer which is attached to the P2 Solo, it meets at least the minimum criteria which Oxford Nanopore has, suggested, because we are not giving any computational support here. For P2I, you don't need to think about that because it comes with the platform. So yeah, I mean, it is like a little bit, I'd say, cost effective compared to P2I, but it comes with this kind of additional criteria, which is completely depending on the user, how they are, including that. So this is also, I mean, I think I already shared the slides deck with you, but I'm going to do it once again today. So another, I think, main criteria is once the system is on, just wait for a minute, and we need to make sure the system, like the entire platform, is initialized properly, and the LED, it should light up once complete, and that would be the indication that it's working well. But I want to also mention this, like I have seen in our New York City lab, because Oxford Nanopore has a research lab, a research and development lab, and also like our office in New York City. And I have seen sometimes this LED light is not working properly. Let's say it's giving yellow color, but actually the system is up and running. So I hope that's not going to happen to you. But if it happens, like I'm just giving you an example, don't rely too much on this. Just let's see like what the software is telling. If there is an error, it will show. If it is not showing, then it's fine. Yeah. Yeah, I think so for today. Okay, I think another quickly, because we are dealing with CFDNA. Yeah, I think this is, once again, as I mentioned, like all of our benchmarking data or the documentation which we have for CFDNA, at least till today, it's based on the plasma extracted CFDNA, not CSF based CFDNA. And as you know, like, when we do some kind of fragmentation analysis, we'd be able to see different peaks because of the association of nucleosome molecule, and I think you're quite familiar with that. So we would be able to see distinct peak for the mononucleosome, dinucleosome, and the polynucleosome. Like, and Cape Station is a good way to do the analysis. Femtopulse is the best because it's way more sensitive than tape station. But if in future, if you do tape station, you can, you know, make sure like you have a tape which has high molecular weight marker. Not D1000 is probably not the best suited for cfDNA. And also now they have a specific tape for cfDNA. I do not know the price difference, but this is what I found yesterday that Agilent now selling something very specific for cfDNA. And they are, the resolution at higher molecular weight is way better than D1000. And yeah, so this is what we suggest, the Kaizen mini elude, but once again, this is based on the plasma cfDNA. And we do also have a protocol for storage of the samples, but once again, not CSF cfDNA. I have found few papers like, when I was checking regarding the CSF cfDNA and Nanopore sequencing. I am going to share those with you. Maybe it could be useful, or maybe you already have the idea of those publications. So I think for now this is what we are gonna stop sharing now. We should start working on our experiment, and then we can come back and finish the rest of the presentation.
Like we were discussing about the genomic contamination and like how we can get rid of it. One of the ways we were thinking about is price selection.
Yeah, I
do you
can
think
do that.
Yeah, okay.
I cannot guarantee whether it's going to help because, this is something I am doing for the first time.
Yeah.
Nanopore has not done CSF, cfDNA. So from Nanopore's side, I don't think we can guarantee anything. But if I think about it, I feel like I would try that.
Okay.
Yeah. Maybe it will help, but not sure. Like, we need to this is the thing, whenever we are dealing with something very new, there could be some optimization steps involved. I'll also try to do more research on it. And so we'll see if someone has done cfDNA, CSF. All the labs, there are like multiple labs who are doing cfDNA sequencing, but none of those labs do CSF, so because the samples are very different, so it could be a very different outcome, but we need to try. I think that is the way I would go as well, because it makes more sense.
Like so that we don't overwhelm the system with and we get our specific.
So yesterday I think I shared like the protocol, right? Which is the cfDNA specific protocol. This one. So we'll do that. So I think the first thing we should do is take out all the reagents, and let's it because it takes time. I just really left the Zoom call. I should join again. You don't have that. You are in the Zoom, Terry?
I can be for the protocol.
Yeah, so if you just send the protocol. Yeah, so if we go to the steps in the workflow,
Overview protocol.
yeah, let's overview. Go, yeah. So here, so we'll decide what we want to do, how many samples we want to pull, based on the QC you have. So the first step would be DNA repair and end prep. So it's kind of more than 2 hours. And then once that is done, that would be the ligation of, the barcode ligation, that is 1 hour. And then, that after ligation and cleanup. That is the final step, that is also close to an hour. And then we can do priming and loading of the flow cell. Okay. So, okay, if we go to like the scroll down, sequencing and analysis, not this here. Compatibility, not equipment and consumables. So yeah, there are like a lot of consumables, right? Other than Nanopore consumables. Let's start, thawing everything. And then you have qubit, right?
Yes.
Okay. And also, I think the magnetic bits are in the kit. And once again, please scroll down. Yeah, so scroll down. Let's go to the, yeah. So because we are doing the native barcoding, I think you have enough material for that. So the material should be okay. And we just need few nanograms. I think 6 nanograms or something per sample. So that should be okay. And when we start also doing this native barcoding protocol, like the end repair, once it is going on, we should also do the flow cell check and want to make sure it passes our warranty. And always whenever flow cell comes, just do a flow cell check. If warranty, like if number of pores are low, it will show that. Then please do it one more time and make sure that is the case. If it happens, email me or email Sam. We would be able to replace it if it is below our warranty, but it has to be within that time period, which is 12 weeks. Flow cell storage, I think that is something very important. Never store it in -20 or at the back of 4 degree. If you do it, that's it. The flow cells would be dead. So that is very important. Okay. Usually at the door of the 4 degree, that's the safest place to store these flow cells. Is it stored properly or not?
Yeah.
They're on a shelf, but they're in the front.
Yeah, there is
The front is fine. Yeah, the front is fine. Yeah, sometimes at 4 degree at the back, the temperature drops, and that is why always at the front is the safe way to go. And never freeze it. That's definitely a no. Okay, so let's scroll down a little bit. Just want to see all the steps. Yeah. What we have to do. So we are done with the sample extraction method. So we'll directly go to the DNA repair. Okay, so this is the DNA. Prepare your cfDNA samples. So if it is concentrated, we have to make sure whatever the proper concentration we need. Just go to number 2. Yeah, let's see here. So 6 nanograms should be minimum, and I think you have 20. So we can just do probably 20 nanograms or something, or 12 nanograms just to be on the safe side. And then this is this is end repair once again. It's an enzymatic step, very straightforward. And yeah,
so you can make master mix for these, right? Based on your sample?
I think some master mix should be okay. I mean, that's what I did in lab,
Okay.
Master mix should be okay. But sometimes, in the protocol, if it says don't do it,
Yeah.
don't do it. But
Okay.
for this one, yeah, it's just entry
Yeah.
there. It should be okay. And yeah, so it is a 15 minutes, and then we are adding the protein is K, something like that. And then again 15 minutes, 5 minutes. So it's like short thermocycler.
Yeah.
And you have like thermocycler
Yeah.
and everything.
We've had all
Right.
these
Yeah.
Okay.
programs.
And then we are adding another end repair enzyme mix. And yeah, say incubate 20-degree 30 minutes, 65. So this is an 1 hour step, right? And then we have the ampere vortexing. So during this 1 hour step, we can do the rest of the presentation, which I would like to explain today. And then there are like before, once after this, we are directly going to the ligation of the barcoding step. And we should have some time in between to take a break and go for lunch. So maybe 1 hour lunch today, that would be permittable. Yeah, I think so. Let's get going. Let's go to the lunch. And once we start pouring all the reagents, we can figure out which samples we want to pull. So we do have a keep up document for the base calling. I am going to share that to you, but that keep up document is more, I think, towards other platforms because we have a fixed computational or a computing part there, right? So what does it mean? It says like, let's say whether you can do 2 soup calling at the same time, or like it has to be run in fast mode or hack mode. And okay, I want to once again go back because I did not today did not talk about like the base calling models, right? Just briefly I mentioned like how the base calling works. So we do have 3 base calling models. One is called fast, which is like I think as the name suggests, although it is an acronym, so it is like, needs less computational power. And then we have a hack model, and then we have a soup model. Soup is the highest accuracy model. So, and the keep up document has mentioned like, let's say you have P24, okay, where there are 24 slots, but that does not mean you can run 24 soup at the same time because it will crash. So that's why that is a very good, I would say, reference when we do base calling, whether it is possible or not. Okay. But unfortunately for P2 Solo, because the computing part is not like it's variable, right? We do not have a straightforward keep up model or keep up document. So what we are gonna do, we will run it in a hack mode and we'll see how the data looks like. For Oxford Nanopore, we can always do rebase calling locally using our base calling software. It's called Dorado, standalone Dorado. So we do it quite often. So we will have all the FASTQ or POT5 files after we do the sequencing. And if we think like we want to run a rebase call using soup model, we can just do it locally. And that is always possible. And that is a good, I think sometimes it is a good strategy to do that way, especially when we are optimizing something like as unique sample as you have. It's always good to do it in a hack mode and then rebase call in soup and see like if there is any difference. Yeah, so this is something I thought of mentioning that for P2 Solo, we'll see which way we can do the base calling or do the sequencing run as well. Okay, so these are the samples, right? Or reagents?
Reagent samples are in 4th degree.
Okay. So I'll just keep it in the IS. And once these are like mass premix are usually all, like enzymes are never frozen. So, um, oh, we need Caesar for honeybee now. It used to have something, right? We
I
don't
know
know
the
that.
little tag.
Yeah. Okay.
Okay.
I think it should be okay. Yeah. Let's just keep another extra one. This is the empty. I'm just gonna set a flash here.
Nah, we can do that later. Yeah. So basically, you just take everything out and put it on ice at the beginning of your day.
Yes, because just to minimize the time of.
Except for these two, which just can be gone.
Because I think these are ampere beads,
Oh.
like spray beads, so it should be fine. And this is a buffer.
Got it.
Yeah, so it should be okay. And we took out one barcode plate. We don't need more than that. I think I have everything. So this, again, keep it at 4 degrees for now.
Okay, and then how about
this
this?
is like, I already took out 3. So everything, for now, let's keep it at 4 degrees. And then, we can keep it at -20 later. I think now we should decide samples.
So, G, if you wouldn't mind that paper at the end, far over.
No, this
is,
yeah.
So that's the printout of the QC if you want to leaf through it briefly, and then I'll put these back and we can talk about them.
Yeah. So let's just see. Also, I want to mention, I think in our documentation for cfDNA, we do not have any specification about what ratio you would expect for cfDNA. For gDNA, they do have.
Nice, yeah.
So I do have some internal cfDNA ratio, which and once again, because although it is an internal, but the sample is different. So this is not like probably an apple to apple comparison, but at least some kind of baseline, I would say. So let me see. So typically 260 over 280, it was between 1.7 to 2.4 plasma cfDNA, and we can actually write it down and mention that it is for a plasma cfDNA. 260 over 280 was 1.7 to 2.4, and 260 to 230, 0.4 to 1. We even with this range, what happens, let's say you have some contaminant present, right? It is for the library prep step, which is the attachment of the adapter. It might hinder it. The efficiency of that enzymatic step could go lower. But for this particular case, it was okay. Everything went properly. And that's why, this is once again not like what we can expect for CSF cfDNA, but I think at least now we have something to compare, some numbers to compare.
These are the internal numbers: 260, 280, 260, 230 plasma. So that's much lower.
Okay. So let's see. And I think this is the newer one, like.
This is all of them. So.
Okay.
And
so we can figure out. So one thing I know, and I think you already know, it's the tape station. I think the most problematic feature, which I thought is like more than these ratios, are the accumulation of high molecular weight fragments, right? That is the, I feel like probably that is the most concerning thing, because it will definitely impact the sequencing run in terms of the coverage per sample. Also, pore blocking may happen. We'll see. I mean, we'll pick the best from here, and then we can think about it, how we can want to run this experiment. So let's
draw your
see.
attention. This one, I think, is our best.
Yeah, it is. Still has something right here.
Correct.
But this peak I can see. This is probably good one. I don't know what. There are two numbers. What are these numbers?
260 over 230 is first.
The first one.
Over the 260.
No, 260 is the second one, right? 0.07.
Yeah.
And this is the.
So number one, number two.
Yeah. So this is actually and this is plasma or like.
This is CSF. The pictures here are just denoting how bloody it is.
Oh, I see.
Because I was trying to correlate between amount of like potential gDNA contamination and bloodiness. As you can see, there's no real correlation though, because this one's very clear.
But still,
and
it is here.
And then you'll note this one is the same actual sample, 2A, 2B, 2C. We just, with protocol modifications, basically more wash steps. And the tape looks more or less uniform throughout, but this one probably looks
this one is the best.
Yeah.
So definitely we are not gonna do number one.
Correct. Yeah.
Number two here, I think we should do it. Yeah, here we do have, because the problem here is maybe a part of it is upper marker, right? But still there are some gDNA like contamination, but hard to say the proportion, like number of in terms of number of moles, because it is not resolved here.
Yeah.
And that's the issue, but let's try it. Number two, I don't think there is anything here.
Yeah,
Right?
What
are these?
This is also the same sample. We tried to run them all on a different extraction protocol, which didn't work.
This one does not work.
Doesn't
Yeah.
work.
So now 2 samples.
I think the next one to draw your attention to would be
Maybe
that one.
this one. Yeah, because these are like definitely not. I don't like this one as well. Here also 1.25.05. I think let's take this. Number 3.
Again, this is the
It's
same
this
sample,
different
different protocols. This would be the most optimized, but again, you can see a ton of.
ton of GDNA here. Yeah. Let's
So,
see like if there is anything else.
No, no, similar to 5. These are all that
No.
protocol. This one was just weird.
What
So
was
these,
this?
A, B, C, these are plasma we tried yesterday,
I see. Yeah.
which we are getting a completely different and weird spectra.
I think here this is upper marker, right?
Yes.
Definitely, but there is something here. So the question here is like maybe because of multiple wash. I don't know what is the
This
difference.
one we only did. This is each individual samples per this exact protocol.
So
no
like
washes.
so this is like not replicates or no wash difference,
No,
different
this is
samples.
Person A, person B, person C.
I see.
Yeah.
Okay. Here, like, the this is the correct peak, I believe, right? This is 100, close to 170 or something, right? Or 150?
They were a little shifted low for the plasma. This is 108,
Oh.
104, 107.
Yeah, plasma it was different. I don't know how. CSF we got it in the correct range, but the plasma, even the nano drop was different. If you look at it, the peak was shifted.
Right, shifted.
Yeah.
Yeah. Which looks like straight. I mean, per the
Yeah.
documents quantities.
Yeah. And yeah, here, like, and sometimes also, I don't know if you have noticed it on tape station. It also depends on the loading. Have you noticed like between different samples the marker gets shifted or it's not calibrated properly?
Yeah,
This is
sometimes-
always my experience with tape station.
Yes, sometimes it.
Also with bioanalyzer, that's what I experienced in the past, and that's I felt like femtopulse is the best, but that is also very expensive equipment, and it's not that we can expect it to have in every lab or something. So yeah, like this is plasma, right? These are plasma again. Yeah. So okay, so 1, 2, 3. So like for today, I think we can just do one plasma pool and one CSF pool, and we'll see.
You are multiplexing it, right?
Yeah, so pool means like here we are picking 3 samples and multiplexing it. And then for the plasma also, I think we should pick 3 samples and multiplex it.
Okay.
And then run like 2 flow cells. Yeah, because once again, this is this is a pilot. Let's say based on the QC, I don't think we should expect like really of high quality data output here because of the presence of the gDNA data would be compromised because it will have some coverage, right? And if number of molecules of the gDNA are more than our actual or like nucleosome peak, then definitely it will get some rates.
So
this is
loading, right? Do you normalize the sample, like the concentration based on the genomic peak that you find, or you just load 6 nanograms in all of the samples?
Oh, so you have to load, I mean, once again, so this is not a, if there is genomic DNA, so if you follow Oxford Nanopore's protocol, they would say we don't have any suggestion, right?
Okay.
Because the way those protocols are optimized is like there should not be any gDNA.
Yeah, okay.
But if you start thinking about it, I would say like we can always normalize based on what is the fraction or like the ratio we are observing in tape station, because maybe we need, let's say for sample A, you have less
1:1
GDNA. Yeah,
ratio.
so we can always, change that ratio. But for the next step would be our end prep, right? For end prep, so let's calculate. So it says at least we need 6 nanogram. So maybe let's, because we have so much contamination and everything, if we have 10 nanogram or 12 nanogram, let's just take that much of sample. Also the volume has to be matched. We have a specific volume for the sample, right?
So you have to dilute it with water?
Let's see, I don't know so the cfDNA, for each reaction it has to be 20 microliters. So if we can just do some quick calculation for 10 nanogram, what is the volume we have? Is it 20 microliter or more? If it is more, we cannot take. We will just take 20 microliter.
Plus minus
Yeah.
20. I mean, we alluded in the recommended 23. So say we use 2 for tea.
Yeah, but what is the concentration? It's more, right? So that's the
They're
thing.
listed here, so it depends. So for the ones we want to do 8 per
So this
microliter.
per microliter 8. So if we take 20, so that's like a really
That's a lot.
lots a lot. That's why maybe for this one we can dilute it. So that's what I
One
mean.
question with respect to the pooling, do you need to normalize the amount per sample, or can you have different? In other words, could I have one that has 10 nanograms, one
can
that has
have
6
it.
nanograms?
Yeah.
You can.
You can have it. I don't think it depends on, once again, so finally it will change in the sample won't be evenly distributed in terms of number of nanograms, but our target is not nanograms. Our target is like to get more reads for like whatever mononucleosome peak we have, right? So if the sample is clean, we don't need to calculate that way. But if the sample is not clean and you want to change the ratio based on that peak versus the GDN, you can always do that. You know what I mean? Yeah.
So I have one more question.
Sure.
Sorry.
No.
Like when you are pooling samples together, right? So you're barcoding them as well? Like
So yeah, if you don't barcode, then you.
yeah, exactly. That's I wanted to know, when do you barcode?
So after this step, so the first step is end repair. The next step would be barcoding, and then the third step is addition of the addition of the protein helicase adapter.
Okay.
And that is the when you do that addition, that's the library that is done.
Okay, that's done.
Yeah.
That's easy.
And yeah, it is easy. It is, but this is still I would say the lengthiest protocol, right? Because of all the enzymatic steps and then purification and because of that. So let's do one thing. Let's however you wanna pull it, calculate. There are 6 samples. So for each reaction, which is the end repair, we need 20 microliter of sample. But I think you need to do dilution of few samples. So you can just do that calculation. Then we can make the master mix and we can start the end repair reaction.
Do you want to briefly go through which 3 plasma we want?
Oh yeah, so because all the samples looks quite, I don't know which one to pick based on what, because everyone has like a
2 weird peaks.
Yeah, exactly. But looks like this one, this is the upper marker, right?
Correct.
And this is, let's say, this is the mononucleosome peak, although it is not the correct, yeah, right? But most likely this is what it is. It could be that Hep station weirdness. Then I would definitely prefer this sample because it has that high molecular weight peak, which is probably less number of moles. Less number of moles compared to this one because this is also higher molecular weight. So this is definitely a good one. Plasma D. Maybe this one as well.
B is in boy.
Yeah, B. What could be the third one? That's the question here. This is C. you can take plasma E, but maybe you can evenly distribute it for today.
Yeah.
But here, like, yeah, it has a higher contamination. So if we want to spike in less number of molecules for this one, we can do it, but we can adjust it later as well.
Okay.
Yeah.
I'll calculate these. Also, your
Um,
paper did indeed come.
It did?
Yeah.
Okay, it was not in a box. Okay,
Yeah,
that's
package.
why. So here, so we are gonna do two pooling. So 3 CSF samples and 3 like plasma. I was not sure like if it was visible to you. So this CSF 01, 02, and 04. And the way I think we are deciding is based on which has the least amount of gDNA contamination, because none of the samples are like, super clean. Every sample has gDNA. And these like high molecular weight peaks, mostly gDNA.
Okay.
I don't think those are the, the polynucleosome, then it should not be this much. Hard to say from,
What about this one? That doesn't have.
it does not have like anything in the correct region.
Oh, okay.
So it's probably the sample, I think, completely lost.
Okay.
For the plasma, we are taking plasma C. We are taking plasma D and E.
Okay.
Even though it has.
Okay.
So maybe, when we are pooling all the sample, we can use less amount of this sample. So maybe the pool we can adjust in such a way it is not evenly distributed.
Okay.
Because if we evenly distribute it, it's fine for a pilot, but we expect there would be a lot of reads for this peak, right? Yeah.
Okay.
So I think at least right now, I think my understanding would be sample prep is very much important,
Yeah.
right? Because I know it is, you are not dealing with a very straightforward kind of sample, but yeah, that's optimization of like which purification method for the cfDNA is working the best. I think that optimization is very important. Yeah.
Sulfur DNA plus.
We can do always
You mean
double-sided sprite cleanup, just to get rid of this high molecular weight. Have you done? So it's a ampere beads. So it's something like this,
Right. So yeah.
the magnetic beads. This is also ampere beads. So okay, so let's say when we run any sample like nucleotide sample DNA or RNA in a gel, right? We are able to see different bands based on different molecular weights. And at least the state of the art method was to cut the gel, and then let's say I want 170 nucleotides, and I have 1,000 nucleotide contaminant there. Then we can always run it in a gel and then cut 170 nucleotides and then do the purification of that particular DNA.
Using the sample that was still in the gel?
You can just cut the band, right? Whenever you cut the band, then you have only that particular band in your Eppendorf, and then there is it's called crush and soak method. So because DNA is nucleotide, these are water soluble.
Yeah.
So usually the buffer is always water based, not any organic solvent. So if it's a very, very classic method. You do cut and soak, crush and soak gel purification method, and you can extract that particular DNA. And then finally, what you have to do, either you have to precipitate it out using ethanol, best purification method, or like spin.
Another question. You in the presentation mentioned that the PCR bias, right? But when you run a gel, you have to run the polymerase chain reaction. But
No. So for this, you don't need polymerase chain reaction. Why you think that?
that's probably I don't worry.
No. So if you have enough DNA sample, you don't need PCR. But
Okay.
let's say you are dealing with really low input sample, then you can always. We also have protocol for low input sample. Then you are introducing PCR. You will be able to sequence it. The only thing you will miss is the base modification information because you are introducing the PCR. So the methylation information would be gone.
Yeah, because when I tell you that I used the min iron.
Yeah.
That did when I did the experiment, we did PCR for that because it was a really small.
Yeah, so it's the same reason.
So,
Yeah, if
okay.
you have small, then I think PCR makes more sense. But for these, like no.
Not necessary.
No,
because-
so
okay.
yeah, so the gel bed purification I explained that is kind of a state of the art way. I think lab scientists used to purify DNA, but now we have the beads.
The beads. Okay.
And so if you, let's say you are using Spry or Ampere, so if you go to their website, they have a very nice diagram of what would be the ratio of the beads to the sample.
Okay.
And based on that ratio, we can try to manage this. Let's say separate out like, a steep cut,
And then just have.
just extract the smaller molecular weight and get rid of the larger molecular weight. I would do that way. I mean, this is not possible today because it is a very long day.
Okay.
But if we have time tomorrow, and if those Ampere beads we have it in the lab, at least we can try it, and then next week, you can just run that
Okay.
and see like if the data looking better.
Right.
I think it should look better because if we get rid of
Get rid
this
of the-
contaminant,
okay.
yeah.
Okay.
So I think that would be the way to go.
Okay.
At least, we can try that way. Yeah, I think why not? If we can recover samples, then that would be more
We can
cleaner
try.
sample, right? And then I think that would be the way to go. Yeah, so let's not do it because already we have all the QC done and everything. Because once we recover samples from those magnetic beads, we definitely need to run it on tape station and make sure we have the correct bead. We need to do nanodrop, we need to do qubit,
Yeah.
and then rest of the steps.
The beads comes in the toolkit that or
So this bead we need at the end as well, before we do the motor enzyme addition, right? This is when we need it. So this is I think you should have a separate beads for
like clean up.
cleanup.
Okay.
Yeah.
But that specific one comes in the.
That specific one is nanopore part. Right?
Okay. Okay.
Works right. Less of plasma. I don't know.
Do you have any questions so far?
Yeah. What is that?
This?
This.
Oh, this is the barcode plates. So different barcodes. Because we are doing multiplexing, which means like every sample should have a different identity, and that identity is the barcode.
You can separate.
Otherwise, it would be mixed. Yeah, so this is the barcode plate. This is not, is it not a clinical lab, right? Or this is a clinical lab? I don't know, in some labs, like, I worked with, they have different specification and protocol. So they just use it one time and then they toss it. But if you are part of research lab, you can just use it.
Okay.
Instead of like this from
Yeah.
So this is
So this is for today. So this is cfDNA. You're not doing any RNA.
So
I don't know how Cubic calculates it. That's like on the screen it shows.
just like that's plasma or CSF, you got a cDNA. So if that cDNA get tested with
It's not cDNA. It's DNA.
Yeah, DNA.
Yeah.
They have the DNA, so the test with different method. So got this.
Yeah, they got
Just
this.
wanna understand.
Yeah, so also, when we are doing the PCR, we should, I'll go over the QC, like what are the necessary QC you have to do before you decide to, sequence any sample. And I would never recommend skip those steps, although it looks very, straightforward, but those are very important. It's like nano drop qubit and tape station, but always do those.
I'm referring to like this number.
Okay. Yeah.
Can you say what medical school?
Mm-hmm.
When you're traveling?
Like 4 months back.
Oh, recently.
So it was first year.
Okay.
7 years. Yeah.
Four.
Well, we can check with her if this these are completely made up.
Okay.
Like I
So I would know more of it's actually less of because they that's
right, so one seems to be the best. So that's why it's
but ask questions.
Yeah.
Because
all right. So here's the calculations that we're proposing.
This one's still or not?
No, only the ones that are starred.
All this one.
Starred only. Not X.
It's not a. And do you think this one's still?
Those two. Starred.
so is it 1.8 for the 360?
It-
Yes. Thank you.
It's okay.
So I guess the first question is to confirm I should be using the qubit concentration.
Yes, absolutely.
Okay, great. So here's the qubit concentrations.
Okay.
Here is the adjustable amount of DNA we can want. So in nanograms, how much we want to add, and then here's the volumes that are calculated. So our thought was for the best one, number one, we would do 16 nanograms.
Okay.
Rather arbitrary.
It's okay. That's fine. Let's go with that.
Two, which has some more contamination, we would do half.
Okay.
So 8.
Mm.
And then this 4,
Mm.
this is the highest we can go just because of the limitation of starting concentration.
Okay, that's fine then. Yeah. And this is 10. And yeah. So this is the first step would be the entry pair, right? So let's do it. So I remember every step. I think we are going to lose some samples because it has purification. So these are all lossy steps. So yeah, let's start, working on it because it's a really long incubation. So
Yes. Give you this back, thank you.
Let me do it because I don't want to be.
I don't know, I name it everyone's in a way.
Sorry.
Think we're good? Yeah.
okay, how you wanna do it? Do you wanna make like a master mix? Let's do that, okay? So the first step, once again, let me show you here. So for each sample, we are taking at least 6 nanograms of extracted cfDNA.
Yeah.
And the volume would be 20 microliters. That's the max volume. And okay, and the reaction is DNA repair, right? So cfDNA 20, buffer 3, and DNA repair mix, which is 0.9. What we can do is we can make a master mix for the buffer and the DNA repair and add 3.9 to each sample. We'll just make probably one reaction extra. You want to make the master mix?
Yes, I'm gonna go get the samples real quick. So do you mind making that for mix?
Yeah, so we have 6 reactions, right? So just make it for 6.5 or 7. Let's do it for 7.
Yeah.
So which means DNA repair buffer 2 to be 27.
21 and the fan mix would be 0.9 multiplied by 7. I think.
So here, please.
6.3. Right?
I don't know. Oh, it's nice. So the goal is I'll try my best to start the sequencing today, you know. So I'll definitely try to do everything by 5. So you have 37 degree, like which thermocycler you want to use today?
I can show you.
Let's make it 37 degree. Also, there are like 6 tubes, right? Let's mark it.
It's hard as PCR tubes. They're over there, and we'll just mark and label.
So let's keep it 37 degree.
We also set it up, and you can check 4 steps as they go. So we have like step 5. Set.
So Mani? So honey, right? Honey. So honey,
Yes,
there is some precipitation here.
that's okay. It's a buffer, right?
Yeah.
Yeah, that's
So should I vortex it or
no,
no?
no, just flick the tube and take the buffer. It should be okay.
Okay.
Oh, I believe it's a step 5. 37 for 15 minutes, and then hold it 4.
Let's start it. Okay, yeah, I'll check this. You can go and start labeling all the PCR tubes.
And you don't. So just pipette, right?
Just swipe it a few times. That should be enough. You mixed and the enzyme.
Yes.
Do you have like a, ice or cold
Yeah. For epigraph tube
or TCR tubes?
just I'll put it on ice.
Give it to me.
Oh, sweet.
But this freezes the enzyme. I don't know. When I do 10x, it freezes.
Oh, because it's at.
Yeah.
Sorry. Let's keep it at.
Is another ice bucket just better?
It's okay. Don't worry. This is. I'm not gonna do it. Hey, if your sample is fine, then it's fine.
Yeah, so that one retains some of these enzymes. Even though, like, I don't know, like, even if you don't go the second thing, it still retains them. Which one was it?
Okay. If you can add your samples, these are 3 plasma. These are 3 CSSF.
Sure.
So 1, 2, 3, 1, 2, 3, and then we can just document later.
Yes, that's one. That's two. Okay, got it. Samples. What's the volume?
20.
Oh, no. Me.
Yeah. Can I at least, I know this is enzyme, you're not supposed to vortex it, but can I spin it
You
down?
don't need to spin it down.
Just
Yeah. Okay. Please add all the samples.
Yeah, I need to make the from the calculations. Sure. I think that's good. This will use your.
Yeah, that is not a good one. Which is like I think 2 or 3 microliters right there. Just a lot of water.
Then the water, just to, right?
Yes, you can spray water. Do you have spray water?
On the out, but yeah, see.
That he is the best. Which one? Oh, that's 16.
CSF 04, 16.2, right?
It's 6. Yeah.
16.2. No. Can you change that to a 7? 6. 6. Gonna come back to it. We can go back to this. Right?
Yeah. 6.1.
Yeah. I think. Do you mind making one more label too? Label CSF.
Yes.
Yeah. Thank you. That should be far. Markers are right there.
So which one is it? BSF of 4, right?
Just write CSF3.
So you are here?
I'm on PG. It's in David. Again, not enough volume. Meaning we have to lower that one. Could you make one more tube for plasma too?
Okay. I'll make one.
Thank you.
What should I name it?
Plasma 2.
Okay.
Okay.
Yep. So that should be 20 microliters of each sample.
So I'm directly adding 3.9 of enzyme in each one. Yes, please.
Yeah. So let's start here. Like, so this part, um, this cover, you are gonna move clockwise, and then you would be able to see this inlet port, and it already has some kind of storage buffer, right, inside in the reservoir. So this inlet port is the port where we are gonna load our samples, but it also has a little bit of air bubble.
So this is the inlet one, right?
Here, this one.
Okay.
Yeah, you'd be able to see it, So what the first thing we are gonna do, we are gonna take out, let's say 20 to 30 microliters of the buffer, whichever is inside already, so that the air pocket is gone. So that is the first thing we are gonna do. So the way we do it is, we are gonna take 1 ml pipette tips. Make it to 200 microliters, and then we are gonna take off the buffer, So upwards, then the like the buffer will come inside this tips. So let's see. The way I do it is you just put it here, and then here you can see it's coming up, right? So I'll just do a little bit more than this.
So you take out 200 or you gauge
No,
it?
we just do probably a little bit more. 30 microliters or so. I did 60.
60 microliters.
There is no like exact volume. You can do 20 to 30 microliters. That should be enough. It's just the purpose is to get rid of that air pocket here. Do the same thing. Once again, make it 200.
So to clarify, you're not trying to remove all the buffer, you're just trying to remove the air bubble.
For this one, yes.
Okay.
Only air bubble.
So you do it until you see an air bubble.
You will see, yeah, here you can see the air bubble, like, if you come here, like, I'm not sure, like, if it is visible. So a small tiny air bubble is there, and that's it. That's the one we want to remove. Sometimes probably there is none, but it is a good practice and recommendation to just get rid of few microliter. And then once this part is done, so usually yeah, you would see the liquid is coming up within so between 220 to 30. So 30 microliters when you have it, I think it should be fine. You can do a little bit more than that. That is not a very strict criteria. Okay. So the next part is the priming mix, which we had made. We are going to load here, and we are going to load 500 microliters, and then 5 minutes incubation, and then 500 again. So in total, 1,000. Do you want to do that?
You can do first.
Okay.
And then I'll do it.
So the way I like to do, and it all depends on how you wanna do it. I don't push it. I just dial it down slowly. In that way, there is no we have more control. No air bubble goes inside. And also the way I do it is first I add a 1 drop there, and then slowly dial it down to maybe just, let's say, little bit buffer is left here. Not everything, because if we want to push down everything, then once again there is a chance we are inserting another air bubble. Okay, so few microliters should be left in the pipette tips, and that time you are gonna remove it. So you can do it.
Oh, so you really need this type of pipette then?
Yeah.
You can't have like a walk-in one.
No. So just add a drop, and it is the same inlet over here. 1 drop, and always I feel like if we can hold it straight, then probably we have more control, and don't push it, just dial it down.
Okay, so can I set it on it?
Yeah. Just
Okay, 1 drop.
1 drop, and then okay, I can show probably
Yes.
one. So there is nothing, no air bubble went inside, right? So just make sure we don't have anything here. Okay. This one. So. And see now, a few microliters left, right? Yeah. That should be enough. Just take it out.
What about the then it's still better so that is fine. Like, oh, it's worked out.
What?
No, I
Oh,
thought there was like a
because there was a drop.
yeah.
That's fine, even if you have it. So 5 minutes incubation they suggest, even if it is a little bit longer. That's
So
fine.
after 5 minutes, again 500.
Yeah. Okay. So we are gonna do 2 times. Do you wanna do this one?
I'll try again. Yeah.
Yeah.
You can do the 5 minutes.
Yeah.
I understand now. Thank you for trying.
No worries.
We're trying to use the same one. I think.
Yeah.
Oh, we just have to.
Okay, so
Yeah. I mean, you can push it, but for me, I don't have that much control. Sometimes, sometimes by chance, if this thumbs comes up, then you are creating a reverse pressure, right? And I don't want to do that. So I just making sure like I don't do that. Yeah.
You worry about the bubble.
Yeah, that's why I'm doing that way.
So
I feel more comfortable doing
Yeah,
this
yeah,
way.
okay, okay.
Some people just don't like it. They are using, I mean, it's fine. For me, I
Okay.
feel more comfortable this way.
Okay, thank
Okay, so this is now 5 minutes incubation.
and do we keep it like this or do we flip the little slide?
This is fine.
Okay.
Yeah, and then so
What else
I
do
think the illusion is done. So we can now prepare the loading mix, which would be, yeah, we can go over there. Oh, you are eluting, right? The incubation is done.
Okay, so so you just have to put it over there and just
Do you wanna log in?
Yeah, of course.
when you set it to 500 and then go down, right? Or 200 go down.
No, it's
It's
not
at
200, it's 500.
Yeah.
Okay. Then you do it completely or you
No,
leave
you
a
don't
bit?
do it completely. You just leave few microliters there. Because
Okay.
if you do completely, there would be air bubble.
Okay.
There is a chance that you are introducing that.
Okay.
So just few microliters when it is left, just you can remove it. That's fine.
It's fine?
That's fine. Yeah. Do it with the same technique.
that's fine,
Yeah, you
yeah.
can just mix it again.
Okay.
So plasma CSF or how you wanna do it?
CSF on the left, plasma on the right.
Okay.
Do you wanna do it or I can do one and then
You
you
can
do
do
one. No, probably fine. Let's see. Um, And once, the sample loading is done, we are gonna just cover this, and that is anti-clockwise. Yeah. You do it or you wanna do it? Okay.
Is it okay if I do it?
Of course. Yeah. It's already 200.
Okay.
Or, yeah, it does not say.
And we're here.
12. This.
That's
12. And then we have to put that, the light shield.
perfectly crystallization.
Yeah, it's a
So
little
that's-
bit, it's a little finicky, right? You saw in
side,
the
right?
morning.
Right. Yeah.
Yeah. The other side, we closed it already. Yeah. And then we are gonna start the sequencing run. It says here as well how to do it.
But it also outputs the electrical signal so we can base call later as well. Is that right?
Absolutely. Yeah. So for this protocol, whatever we shared, it is for CFDNA, right? So they are saying to use hack. So I think hack should be enough. Like, high accuracy calling, if that is making any difference.
How do I do it?
Anti-clockwise.
Anti-
This way.
Oh, okay.
Okay, so you can, there is light shield. You want to do it, Terry?
Why do we need the light shield?
I think presently there is no bleaching or anything, and that current chemistry or like so nanopore, like whatever pore you are using now, it is called R10. Previously we had R9, and I think there was some kind of photo bleaching happened. So it is just for the safety, but presently I think they say it is an optional.
Okay.
Yeah, with an R10 chemistry, but it's the recommended one. We'll just do it.
Okay.
Yeah.
Might have to check those. It feels like, they seem good.
Yeah, this is the way.
Okay.
Um,
And they're
so
like
this,
pressed down
yeah,
enough.
I think this one is little bit, I felt it is problematic, but I think it's okay. I think this is how the light shield is made. Yeah, because this is not the smooth side, right?
Correct. That's
Yeah,
rough
so it
side.
is the rough side, and that's how it should be.
So shut
So
this.
shut this, and then you can do it. I can show you. Let's go to start sequencing. We can do some CFDNA pilot, however you want to name it.
So 19th.
You can do ANT because this is a part of the training. Yeah, CFDNA. Select all positions. Okay. Let's do sample ID as well, which is however you want to call it. So it was CSF and plasma.
Correct. So A is the left.
Yeah.
Okay. So meaning this is left CSF.
Mm-hmm.
So I want CSF. Oh, I guess.
Any other, extension you want to add for your sample identification, you can do it.
No. Unless you think I should add more. Because
Okay.
it'll always be
Do you wanna?
under here,
Yeah,
right?
it should be. Do you wanna add just for our own record the barcode? So
Yes, please.
So I think the CSF one was 1 to 3, right? And plasma one 4 to 6.
And I guess they were all A.
What do you mean like?
In the barcode plate.
So
we
it's the
started
first
on A1,
column.
so A1, A2, A3, A4, A5, A6, rather than going across.